Welcome everyone to this week's edition of the Extracellular Vesicle Club. We have a topic, as we were just discussing, that's of great interest to a lot of people because many of us are working with samples from patients, and most of those who are working with those samples are working with blood, so blood products like plasma or serum. So we're going to be hearing a bit today about different separation methods for plasma EVs, blood plasma EVs, and also about some omics approaches to characterizing those EVs. And today's EV Club is a collaboration with the Student Network on EVs. I'm going to put the website into the chat box here, but we have a special guest moderator today, Nazanin Rahmani. So Nazanin, thank you so much for joining and for agreeing to host with me today. And I'd just like to remind everybody to please put your comments and questions in the chat box. And then at the end of Daan's presentation, we can get to you in order. So Nazanin, can you tell us a bit about SNEV and then introduce our speaker today?
Hello, everyone. Thank you so much, Ken, for giving me this opportunity, and welcome everyone to this session of the AV Club. It's my pleasure and an honor to have me here. As mentioned, I'm Nazanin Rahmani. I recently earned my bachelor in cell and molecular biology, and I'm currently applying for graduate studies and an executive member of SNEV, the Student Network on Extracellular Vesicles. SNEV offers a platform of the students to discuss, share knowledge, network, and collaborate with other students and early career researchers in the field of extracellular vesicles. And I like to say that I became interested in EVs when I was a sophomore, and that's when I joined SNEV. Being a part of SNEV has been immensely helpful in my academic journey, and it has allowed me to connect with senior students in the field. I remember I was participating EV Club meetings during that time, and I was always nervous to ask questions in the presence of PhD students. Also now nervous as well. I just want to say that if there is anyone else in the same situation as me, I can confidently say that SNEV can be the best platform. Because we have various events where we not only focus on scientific knowledge, but also soft skills. These activities take place virtually, so there are no restrictions based on your location. Additionally, SNEV is about to hold its second virtual conference. Unfortunately, the abstract submission process has closed, but you can still register to attend the conference. We have fantastic keynote speakers, presentations, and so on. All right, today we have the pleasure, and it's my honor to hosting Daan Paget, a PhD student at University of Oxford studying pharmacology and cardiovascular medicine. His publication is about comparative and integrated analysis of plasma extracellular vesicles isolation methods in healthy volunteers and patients following myocardial infarction. Hello, Daan. I'd like to invite you to share your screen, and I would appreciate if you introduce yourself again and then begin your presentation.
Yeah, just to make sure, I am sharing my screen now, right?
Oh, yeah.
Yeah, perfect. Yeah, so thank you for the lovely introduction and the opportunity to speak at the EV Club. And so, yeah, my name is Daan Paget, and today I'll share data on my PhD work where I looked at the integrated omic plasma extracellular vesicle profile and how it's influenced by the isolation method in both healthy volunteers and myocardial infarction patients. And so this is work that was published less than a year ago, and at the end of the presentation, I'll share some new data just to spice things up. So yeah, to get started, for the people that are not in cardiovascular medicine, a myocardial infarction, also known as a heart attack, is essentially caused by a atherosclerotic plaque that ruptures and then causes an occlusion in the coronary artery, which then leads to ischemic damage to the heart, and then leads to a large inflammatory process. And so, of course, when we look at the plasma of patients in this following a myocardial infarction, we find extracellular vesicles. And so previously, the group I work in Oxford it's found that at time of presentation, so in this very acute phase following the myocardial infarction, we see significantly more extracellular vesicles present than the same patient 6 months later. And so when I talk about the acute phase, I mean within 2 hours after the heart attack occurring. And so what's interesting is that we then found that the amount of extracellular vesicles at this acute phase correlates with the extent of injury. So the larger the infarct of the patient, the more extracellular vesicles are present at this acute time point. And so this very nicely shows how plasma extracellular vesicles can function as a biomarker in cardiovascular disease. And so we know that recent studies have looked more into characterizing the extracellular vesicles more in depth by looking at their cargo, by both doing lipidomic and proteomic approaches, looking at the protein profile of these extracellular vesicles and the lipid profile of these extracellular vesicles. And so we wanted to be one of the first studies to look at both the protein profile and the lipid profile of extracellular vesicles following a myocardial infarction. And the reason why we're looking at both is we want to get a total picture of the extracellular vesicle profile or signature. However, we know that when we work on plasma, one of the main challenges we face is that there are a lot of contaminants present in the plasma that are co-isolated with extracellular vesicles. So here we can see plasma proteins such as albumin and fibrinogen, and lipoproteins such as HDL and LDL that can masquerade the extracellular vesicle profile. So before undertaking this big study of looking at extracellular vesicles following myocardial infarction and looking at their omics profile, we first set out to see how the plasma EV isolation method affects the EV profile, and then to characterize the extracellular vesicle profile following a myocardial infarction, and seeing how the isolation method affects the ability to see clinical alterations between the two different profiles. So perfect. So to address the first question, we first wanted to see how the isolation method affects the EV omic acquired profile that we see. And so we started relatively simple by just using healthy volunteers, taking plasma, and then isolating extracellular vesicles using 5 different methods. So we use ultracentrifugation, which is the golden standard or is the most heavily used method. We use precipitation, which is a method based on PEG polymer precipitation of extracellular vesicles directly from plasma. Acoustic trapping, which is a more novel approach by a company in Sweden, where they use sound waves to isolate extracellular vesicles directly from plasma. Size exclusion chromatography, which isolates EVs based on size. And then lastly, we use immunoaffinity capture versus common extracellular vesicle markers, the tetraspanins, CD9, CD63, and CD81. And so the idea there is we have magnetic beads that we then coat with these antibodies and then pull down extracellular vesicles from plasma. And what we found was that in other studies where they used this approach, we were missing an IgG control, and so we used a matched IgG control. And so another important thing to note is that all the data I'm sharing here is a delta between the platelets for plasma versus the PBS to remove any method contaminants that are present that can interfere with the EV profile. And so following EV isolation, we then characterize the extracellular vesicles in depth by looking at their size and number using nanoparticle tracking analysis, confirming we had extracellular vesicles using Western blot and transmissional electron microscopy, and then looking at the protein profile using an EV array analysis, which I'll explain later, unbiased proteomics, and then looking at the lipid profile by doing a targeted sphingolipidomic analysis. And so following establishing this profile, we then combined all the data and we did a principal components analysis to look at the total EV profile in its whole. And so the way it will work is I'll first talk you through the individual characterization data points and then go into the integrated data set. And so we first started by looking at the particle concentration using nanoparticle tracking analysis. And what we find is when we compare the different isolation methods is that precipitation-based isolation method yields the highest particle concentration compared to the other isolation methods, where we see about 100-fold increase in the number of particles present. And we then looked at the size and the size distribution profile. And what you can see here nicely is that there's not really a huge difference if we look at the size distribution profile between the different isolation methods. And you might notice that immunoaffinity capture currently is missing, and that's mainly because it's hard to measure the size, or actually impossible to measure the size without eluting the extracellular vesicles, which can affect the size in the first place, because usually you use a quite strong acid to elute them. And so similar to what we see in the particle concentration, when we look at the protein concentration using a bichronic acid assay, we see again that precipitation yields this very large particle count or protein concentration, indicating that most likely it's quite impure, or it just seems to be very efficient at isolating extracellular vesicles. But we know from other publications that it's most likely impurities that we're seeing. So great. So following the typical NTA data analysis, we then looked at Western blot to confirm that we had extracellular vesicles present. And so we looked at both plasma and PBS control to see if there were a signature from the method itself. And that was mainly a concern with precipitation and, for example, acoustic trapping. And so then we isolated the extracellular vesicles and looked at two extracellular vesicle markers, being ALIX and CD63. And so we all know that blotting for extracellular vesicles is quite a challenge. And so here what you can see are two Western blots where we can see that ALIX and CD63, which are EV markers, are enriched in the plasma sections, but we see no signal if we look at the PBS control. And we see that for all these four methods here. So UC, precipitation, acoustic trapping, and SEC. And then if we look at immunoaffinity capture, one thing that I'd like to point out in particular is that for the IgG beads, so the control beads, we still see ALIX and CD63 present in these samples, indicating that there's some non-specific binding occurring. And so then besides looking at extracellular vesicle markers, we then want to look at these common plasma contaminants. And so we looked at ApoB, albumin, and ApoA1, which are indicative of plasma proteins and lipoproteins. And again, we see that in all isolation methods, even the immunoaffinity capture method, we see this strong contaminant presence, which you can see in the blots here. Perfect. So following confirmation that we seem to have extracellular vesicles present based on the Western blots, we then want to image the extracellular vesicles using transmission electron microscopy. And so again, if we look at the four more basic methods, ultracentrifugation, precipitation, acoustic trapping, and SEC, here we can see nicely on the TEM images that we do have EV-like particles present, which is great. And then we face the challenge that when we want to image the beads sections, we have to use a slightly different approach. And so what we did was we took the magnetic beads with the extracellular vesicles bound, we then fixed them into acheros, and then sliced them into small cubes and imaged the cubes independently. And so what you can see here is the bead. So I'm going to get the laser pointer out, but you can see here the magnetic beads. And if we zoom in on the surface of the beads, we see no extracellular vesicle present for the IgG beads. But when we look at the tetraspanin beads, so CD9, CD81, and CD63, we see that extracellular vesicle-like particles are present at the surface of the beads, nicely showing that we are isolating EVs. So following the baseline characterization, following the ICIF guidelines, we then wanted to do a more in-depth look into the protein profile and how the isolation method affects this protein profile. And so we first started by doing a targeted protein analysis using the EV array. And so this was a collaboration with Aalborg, with Rikke Beek and Milena Jurgensen. And essentially what they have is this a device which can image extracellular vesicles and quantify the protein. And so here we can see how it works. We have this chip that is coated with antibodies that target extracellular vesicles. And then once the extracellular vesicles are bound, you can then chase the extracellular vesicles with these labeled antibodies and quantify the proteins. And so we first started quite modest and we just looked at extracellular vesicle markers. And already we can see that the isolation method affects the protein profile that we see, where we can see that CD9 and CD81 are significantly higher in ultracentrifugation and precipitation when we compare them to other methods. And these difference in the protein profile becomes more apparent if we expand this panel looking at cell-associated markers. We wanted to see what kind of cell-associated extracellular vesicles were enriched. And here you can see a panel of these markers based on the EV array. And so, for example, when we look at CD41, which is the significant one in the approximately in the middle, we can very nicely see a strong CD41 positive extracellular vesicle subpopulation in ultracentrifugation isolated samples. But when we look at SEC, for example, we don't really see this enrichment. And so to further confirm this, we then did unbiased proteomics to look at the total protein profile of these extracellular vesicles and to see how the isolation method affects this protein profile. And so what you can see here is a heat map of the top 30 proteins common amongst all methods. And so as you can see here you can see the top 30 proteins, and at the bottom you can see the different isolation methods. And the first thing I'd like to point out is that we see very nice clustering of patients or healthy volunteers together. And what we can then see is that when we apply a hierarchical clustering to see if there are differences between these different groups, we see that there are two main clusters forming, which is SEC and UC, which seem to be quite high in these proteins, and then immunoaffinity capture, acoustic trapping, and precipitation, which seems to have a lower amount of these proteins present. So again, suggesting that the protein profile is affected by the isolation method. So we then moved into a lipidomic approach. And so what we decided to do is look specifically at sphingolipids as the most enriched lipid in extracellular vesicles. And so this study in plasma extracellular vesicles comparing isolation method hasn't been done before. So we were quite excited to see what we got. And interestingly, what we see when we look at the data is we see that there is a strong difference in the lipid signature comparing the different isolation methods. And in particular, precipitation seems to be very lipid enriched. Where here, if we look at the precipitation area, we see a strong enrichment in, for example, different ceramides and sphingomyelins as well. And when we look at the other isolation methods, we see that they cluster nicely together, and we don't really see a very strong lipid signature present. So great, so we see differences in the protein concentration, we see differences in the particle concentration, we see differences in the protein profile and the lipid profile. What happens if we integrate all this data together and look at the total picture? And so to do that, we use principal component analysis, which essentially is a way to reduce the dimensionality of your data. And and essentially what it does is it takes your 1,000 data points that you feed it, and it then builds these conglomerates or these combinations of different data set points to separate the data. And so when we do that, and we look at the principal components analysis, we see again that there's a big difference in the, a big difference between different isolation methods, where we can see that precipitation, ultracentrifugation, and SEC form these separate distinct clusters, and acoustic trapping and immunoaffinity capture cluster together. And so just to talk you through a principal component plot, essentially PC1 mainly consists of proteomic data, and then principal component 2 mainly consists of lipidomic data. The individual points that you see here are individual healthy volunteers, and when we look at these circles, those indicate the 95% confidence intervals. So great, we see that the isolation method affects the omics acquired profile that we see. And so we then want to move into our second question, which is looking at the EV profile following a myocardial infarction and looking how the isolation method affects the ability to see clinical differences between patient groups. And so to do that, we used a very nice study being ran in Oxford right now, which is called OXFAMI. And so OXFAMI is essentially a study looking at patients following a myocardial infarction, where we get blood from patients within 2 hours following the infarct, so in this very acute phase, and then we follow this patient and we get these highly characterized patients throughout. So we have a blood points at 2 hours and then at 1 month, and then we have cardiac MRI imaging of the same patient at 6 months. And so as you can see here, the patients were about 67 years old. They were only male patients because we wanted to remove any sex differences. And then you can see that the infarct they had at 6 months was approximately 15%, which is relatively large, yeah. And so then following this study, we then did the same pipeline where we isolated the extracellular vesicles using 5 different methods, then characterized them, and then looked at the protein profile and lipid profile. And so again, I'll talk you through the data points one by one, and we can see how what the difference is between the EV profile time of presentation versus the same patient 1 month later. Again, we started with nanoparticle tracking analysis, where we first wanted to see if we found similar results to data that we published previously. And so previously what we found was that there was a significant higher amounts of extracellular vesicles present at time of presentation versus follow-up. And in this similar patient cohort, we again find the same thing when we use the same isolation technique, which is ultracentrifugation. And interestingly, when we apply the same analysis to the other isolation methods, we see a similar trend where we see more extracellular vesicles at this acute time point versus the same patients 1 month later. And so then we wanted to see if we saw a nice correlation, which we saw previously with the infarct size. And so we looked at cardiac MRI imaging in these patients and then compared the plasma concentration at time of presentation. And what we see is we see a nice correlation when we look at ultracentrifugation isolated extracellular vesicles, where the larger the infarct, the more extracellular vesicles are present. But what's interesting is if we then apply the same analysis to other EV isolation methods, we don't find any correlation at all. And so again, this suggests that the isolation method does affect the profile that you're seeing. And so moving forward from the data, we then wanted to look at the protein data. And so we did the EV array analysis in these samples, and we expressed the data of fold at time of presentation versus the same patient at 1 month follow-up. And again, when we look at the extracellular vesicle markers, we find that there are quite distinct differences between the different isolation groups, where red in this graph indicates, or this heat map indicates, an increase at time of presentation, whereas blue indicates a decrease. And so we can see that there is intervariability between patients. And again, we can kind of see if we look at the global profile that there are differences between the different isolation methods, but this becomes even more apparent if we look at cell-associated markers, where we can see big differences between the different groups. And again, this is a bit difficult to look at. So when we zoom in on one particular one, CD31, which is an endothelial cell marker, we see, for example, that at ultracentrifugation, there's an increase at time of presentation versus 1 month follow-up. Whereas if we isolate the EVs using acoustic trapping or SEC, we see a general decline or a decrease of CD31 positive extracellular vesicles. Again, suggesting that there are differences between the patient group at time of presentation versus follow-up, and that the isolation method affects this difference. So we then did the sphingolipid analysis. So we took the patients again at time of presentation and follow-up, isolated the EVs, and looked at the sphingolipid concentration and expressed it again as fold of enrichments versus 1 month follow-up. And what we see again is a very stark difference between the different isolation methods. So in particular, precipitation seems to have a huge increase at time of presentation versus the 1 month follow-up, whereas for other methods, this is less emphasized. And again, when we zoom in on, for example, ceramide 24, we see a big difference. So ceramide 24 for ultracentrifugation is equal between time of presentation and 1 month follow-up. But then when we isolate extracellular vesicles using precipitation, we see about a 3-fold increase at this acute phase. So this was very interesting data. Yeah. Cool. So then we see differences in the concentration, we see differences in the protein profile, we see differences in the lipid profile of these MI patients. And so what happens if we integrate all this data together, and are we then able to differentiate between a patient sample at time of presentation, so in the acute stress phase, versus a patient sample at a 1 month follow-up? And the answer is essentially that only 2 methods were able to differentiate it based on the EV profile that we obtained. So here you can see the 5 different isolation methods, and you can see a principal component analysis where we try to separate the EV profile time of presentation versus the EV profile in the same patient 1 month follow-up. And only 2 methods were able to clearly separate the 2 groups. which you can see by looking at the 95% interval ovals, essentially. And so if they overlap, it means we couldn't really see a difference in the EV profile. But if we see a nice separation, we can see that there is a difference between these two profiles. And so this was quite interesting to look at. And so essentially, this is kind of where the paper ends. And so what we found in conclusion of this data was that each isolation method contains a core EV signature, which is all data that I kind of left in the supplementary data. But essentially, we compared our data with previously published studies and found a strong overlap between our data and others. We then also found that the isolation method affects the plasma EV profile. And we then showed that the EV isolation method influences biomarker discovery in MI patients, as only 2 methods were able to differentiate between EV profiles. And so, yeah, that's where the paper ends, but that's not really where the story ends. And so we did a further in-depth analysis. And what we wanted to see is if we look at all the patients and all the isolation methods, what are the common differentiators? So if we look across all the methods, what are the main things that are differentiating the acute phase MI profile versus the follow-up MI profile? And unfortunately, because this is work in progress, I can't really show the main markers, but what is interesting is that it mainly seems to be an endothelial cell-derived EV profile that is driving this difference between presentation and follow-up. And so this is work that we're currently working on right now, where we're looking more in depth in why we're seeing this endothelial cell-derived EV response. Similarly, because we have healthy volunteer data and MI data, we wanted to see, well, what happens if we compare the two? Can we differentiate between a healthy person, an acute phase MI patient, and a 1-month follow-up MI patient? And so here you can see what that looks like for acoustic trapping, where we can see a clear separation between the 3 groups. So we have our healthy volunteers on the left, then we have our presentation at the top right, and then the 1-month follow-up patient in the bottom right. And so we can see a clear difference. And when we look at the key common denominators between these groups, we can find that it's mainly based on the lipid profile. So it seems to suggest that the lipid profile is differentiating these patients. And so the reason why, for example, here you can see the direction of change alters is because based on the method, we've seen increase or decrease in the MI patients versus the healthy volunteers. Just to add to the conclusion, we found our 3 previous conclusions, and then we also found that the common subpopulation we see following an MI seems to be from endothelial cells. And so with that, I would like to thank everyone for listening. I would like to welcome you for a discussion, and I'd like to thank all my supervisors and collaborators. Any questions, please let me know.
Thank you so much, Dan. It was a perfect presentation and really educational for me. If anyone has a question, you can ask in a chat box, or you can open your mic or cam and ask yourself. Until then, you can answer the questions in the chat box. They asked, what is the average number of EVs in plasma, and what would you say is the average protein yield per given volume of plasma serving as EV source?
Yeah, so I think that's a good question. So actually, there's a lot of preliminary data that I didn't really show that built up to this study. And so initially, we started with how much plasma do we even need to put as an input to get a nice signature out of it. And so for the healthy volunteer data, we used 500 microliters of plasma. But then for the patient samples, we were able to get a nice signal with 100 microliters of plasma. And so what we see again is depending on the patients, depending on the fasting state, depending on many, many factors, we see about 10 to the 10th or 10 to the 12th, depending on isolation method, of extracellular vesicles present. And again, when we say, actually, I should say particles present, because with NTA, and I saw a question actually touching on this in the chat box, but with NTA, we only measure particles, not really extracellular vesicles. So that's an important differentiation to make. Yeah. So yeah, again, about 100 microliters should be sufficient with these isolation methods. Yeah.
Michael and Maria asked, what was the starting volume of blood for each of the isolation methods?
Yeah, so again, I think, so I kind of answered that, but just to clarify is we use the same starting volume for every single method. And so you can normalize based on many things, but we decided to normalize on the starting inputs. So again, for healthy volunteers, it was 500 microliters. And for the MI patients, it was 100 microliters, seeing as the samples were a bit more scarce. And so the reason actually we also used 100 microliters is because this was designed for a very large patient database. And so we wanted to see, can we use as little as possible? And the answer is 100 is perfect, especially looking at lipids, because at some point, if you use lower amounts, you start struggling to get a good signature for proteomics, but also for sphingolipidomics.
Yeah. How do you know that you are not measuring LDL and HDL related proteins and sphingolipids from lipoprotein particles, which are still a very disturbing factors in plasma EV research?
Yeah, a great question. And I mean, that is actually a big issue. So essentially, as I said, NTA doesn't differentiate between a random particle or something that's about 100 nanometers of size versus extracellular vesicles. And so we contemplated using, for example, FACS, where you can label your solution with CD9 or other tetraspanons that are EV positive and doing the analysis like that. But unfortunately, it was essentially would require a lot more experimentation. And so at some points, you can't really differentiate. And for the sphingolipidomic analysis, it's another great point. We're not sure if the sphingolipids we're measuring are actually from the extracellular vesicles or if they're from lipoproteins. And that's something that, yeah, you would either need to have a really pure isolation method to then look at the specific signal, which would require a ton of plasma inputs. Or you need to do something with like lipid labeling or something, or like, yeah, but that's a tricky challenge. And I think in the lipid field, it's not really established a good protocol to do that. Yeah.
Okay, there is a question that for your MT analysis, what did you use to differentiate EV count vs total particle count?
Yeah, again, so the
answered.
in the publication, yeah, I answered that. In the publication, we used particle concentration in previous studies, and actually we have done those studies for not the paper where we label the EVs essentially. Yeah.
Yeah. Have you tried transproteomic analysis, qPCR analysis of microRNA, mRNA?
Yeah. So personally, I would have loved to add this analysis to it, but the paper was already getting big, and there actually have been papers where they've done a transcriptomic analysis comparison. And so one of them, I think that's very interesting, that was published in JEV recently, was by Firman, by Suzanne Gabrielson, which I think was published also last year, where they looked at transcriptomic analysis comparison. But unfortunately, we didn't do those on the samples. But with the future studies that we're planning, that's definitely a consideration we're taking into account. Yeah.
Yeah, they ask, can you speculate how to preserve the corona during enrichment?
Sorry, what was which enrichments?
Corona during.
The corona, the protein corona.
Yeah.
Oh, that's a super interesting question. Okay, yeah. So, of course, I think most of the field now is familiar with the work by Buzas in Semmelweis University, where they look at the protein corona, and they even looked a comparison between SEC and UC, I believe, where they saw some differences in the protein corona forming around the EV. So we don't really know when we do the proteomic analysis here and the protein array analysis, if it's within the EV or on the surface of the EV. So we didn't really differentiate here between that. So that would be a specific analysis that you could do, but it has been found that the isolation method affects that. But one thing in particular I think is very interesting is looking at the lipid corona. And so that's work that hasn't really been done, where they look at how the lipid absorption to extracellular vesicles on the surface is affected, and if that even occurs in the first place. And so that's something that, yeah, it hasn't really been done yet, but would be very interesting to look at. Yeah.
Yeah, they asked, What do you think is responsible for the unspecific binding observed in the IgG pull down? And also, did you include the IgG pull down control in a proteomic analysis?
Yeah, so I'm not sure if that was very clear during my talk then, but so great points. We saw non-specific binding to the IgG beads. And so all the data that was expressed in this manuscript is essentially a delta of the tetraspanin signature versus the IgG signature. So we're trying to correct for that non-specific binding. So what the reason was for this non-specific binding, I think it's like almost sticky extracellular vesicles or something like that that are adhering, or fragments of extracellular vesicles that are adhering to these beads. Mainly because this was also found by a group in Boston from Shannon Stott's lab, where they found that if you run plasma through just an empty chip, they were doing some chip isolations on the extracellular vesicles, you still get sticking to the sides. And so it seems to suggest that there's this like adherent EV subpopulation that seems to bind to IgG beads. And so again, this was not really done in any other plasma research that we found, so we wanted to include it, and I think the result we found was quite interesting there. Yeah.
Were all analyses performed on frozen samples?
Yeah, another great point. So all the samples were performed on frozen samples. The main kind of reason behind that was that this study was kind of meant to be used for like a reference when you do database analysis. So if you have a database of patients, for example, following a stroke, then even if it's frozen, we know that you can get a nice EV signature, a nice lipid signature. And so, yeah, comparing frozen versus fresh, we haven't really done. I have some data on the proteomic parts and on the NTA data, but that was not included in the paper, so no.
Henry asked, did you do NTA analysis at the plasma itself before EV isolation, and how's comparative number of EV population after isolation?
Yeah, that's a great question. So I can say that anyone who owns an NTA device would probably advise against running a pure plasma sample into the machine, because it can damage the machine essentially. So we have done it, but you have to first dilute very heavily, and secondly, it's definitely not advisable. So even, for example, using the precipitation-based method, we were a bit worried about the NTA machine. So I can't really say specifically what the number is, but yeah, that's actually a good question. Yeah.
Yeah. Elena said that she was preserving lipid in the corona.
Oh, so okay, hang on. So what was her question specifically was?
Let me.
It's clear that changing the ratio of sphingomyelin and ceramides can interfere with the protein. Oh, that's a very tough question. Yeah, I mean, so again, that is I think looking at lipid specific signature is not really super heavily studied right now. I know some of the people in the field are moving towards it, which I'm very happy about, but all those kinds of dynamics, it's very hard to say in extracellular vesicle perspective in general. What I can say is when we look at our a similar field, such as the liposome field or the lipid nanoparticle field, it's true that changing the lipid composure of the nanoparticle can affect the protein corona. And also it's true that in that field, you can find lipids binding to the nanoparticles in general. So I think it's definitely occurring, but we just don't know. So, but yeah, it's a really interesting question.
There is a question that is, what is your outlook on using tetraspanin-based immunocapture approaches to isolate EVs from plasma? So
I personally think so. It's a kind of I think it's a known fact in the EV field that it's quite tricky to isolate specific subpopulations or even just with immunoaffinity capture, and it requires a lot of optimization. What I can say for the paper is, based on the method we have here, we do see a signature, which is nice, but I think it would require further optimization essentially. So I would say is it's tricky, definitely, mainly because of this sticky EV population. And so I think it's definitely a good approach because it seems to be quite pure, but there are also some downsides, right? Like eluting the beads, eluting the EVs from the beads can be very difficult. So I know people use like acidic washes, but then of course you can also affect the EV itself. And so what I would say is that one of the issues, for example, is if you want to look at functional changes, you then have to elute the EVs and see if the functional change is even still there. So I think it's good. It's interesting, but it just requires a bit more work in the future, I think. But yeah, that's of course, my 2 cents. It's not on paper somewhere. So yeah.
Yeah. Have you tried to combine 2 different EVs isolation techniques sequentially?
Yeah, I think, again, great question. So there are really lovely papers out there where they use 2 methods, even 3 methods to reach these really pure EV isolations. And it's very interesting. We limited ourselves to a more single step approach because, again, in mind, we had this really big patient cohort data in mind. And so combining different techniques would definitely increase the handling time and is maybe less suitable for these large clinical approaches. But yeah, definitely would probably be a more pure isolation method if you combine these methods. And there are papers on that. Yeah, but good question again.
Yeah. Did you look at the impact of different methods on the mitochondrial proteins in EVs?
Great question. I think it's definitely something the field's moving towards. We haven't looked at mitochondrial proteins, and no. So yeah, that's to be done at some point.
Do you use anything for the cryopreservation of EVs in the plasma samples?
So right now what we do is we essentially spin the blood, remove the platelets from the blood, so from the plasma, sorry, and then we freeze it. And in theory, that should be fine. I mean, we found that it doesn't really affect the signature that much. So yeah.
Yeah, they asked it was increasing use of a TFF as a possible EV isolation method. What was the reason to not include in this study?
Yeah, so actually, if you look at the different EV isolation methods, there are many out there. And so at some point we were just limited. And so we decided to go for like 3 very commonly used ones. So precipitation is very heavily used in clinical labs because it's so easy. Ultracentrifugation, of course, is very heavily used. SEC is heavily used. And then we wanted to use a more different approach, which would be the acoustic trapping. And then lastly, we used immunoaffinity capture because it's kind of heralded as like this very pure method. But yeah, like including more study would be interesting. Yeah. But unfortunately, again, it was kind of past the capabilities. Yeah.
Have you ever noticed decrease in number of EVs after freezing? For how long plasma was stored maximum before collecting EVs?
Yeah, so good question. So I know there are studies on this. I don't have them. I don't know them top of my head, but essentially they were frozen for I think it was 2-3 years or something. And I think there's studies looking at like how plasma samples are affected over time. But I don't have the data there. So I recommend you to go and try and find those studies. So yeah.
There is another question about this. How many freeze thaw cycles is allowed for these samples?
Yeah, so I would heavily recommend to limit because there is data on freeze thaw being like there's a limit essentially on how many times you can freeze thaw the EVs. And so we try to do everything with just 1 freeze thaw essentially. Sure, some of the sample shipments was a bit more difficult, but essentially we tried to limit freeze thaw completely because we didn't want to affect the proteins and lipids in the EVs. Yeah.
What is the starting volume of plasma acquired for each patient?
So that is a good question. So I know we started with 20 mils of blood, 20 to 30 mils of blood because it was just taken in the clinic. And from that you get quite a lot of plasma. I'm not entirely sure how much exactly, but you get like, I think you get like 4 mil from healthy volunteers, if I remember correctly, maybe even 5 mil. Of course, of platelet poor plasma as well, I should add, because yeah, there's a step in between there. Yeah.
Yeah, question finished. If anyone else has any question, we can.
Yeah, that's perfect.
Up.
Yeah.
Hi.
Oh, sorry. Hi.
Hi, Danina here. I have posted the question in the chat box, but perhaps you missed it. That's totally fine. I was just like wondering, like you already covered in your talk the difference between extracellular vesicles as we know them and in general particles. And with the different methodologies that you have applied in your paper, one can conclude that in general, the difference is between the yield and the purity of your preparation, right? And this was the driven factor for the differences that you've seen and how you clustered your samples. And I was wondering, like in the very last slide that you showed with the data that are, with the experiments that are ongoing in your lab, if you plan on validating the EV origin of the biomarkers of interest that you may have identified to specifically link them to EVs and not other particles that you may have in your preps.
Yeah, I think that's a very, very valid remark. So yeah, essentially what I would recommend in general is that once you have your marker of interest, do a very pure method essentially, or maybe combining two methods to see if you still see the marker presence. And so that's something that we are doing. But yeah, again, because we were doing this big comparison with single step, we were limited by that. But I think in general, if you ever find a marker for anything, it's good to do like a more in-depth like characterization to really show, maybe with TEM, like gold labeling, but like, yeah, to show that it's present. Yeah, so very good remark. Yeah.
Not saying that it needs to be linked to EVs, okay? So especially if it's a biomarker, as soon as like it's a stable biomarker across your cohort. But yeah, I was just like curious about that. Great talk. Thank you so much.
Yeah, thanks so much. Yeah.
There's another question that how was the plated pool plasma prepared? How was it?
That I would recommend to check the publication. I know we did a spin on it, but I'm not entirely sure what speed we use. So unfortunately, that's not fresh in my head. And then one question I actually see in particular from Dirk Jan is looking at fasting states, and I think that's an excellent question because there are studies showing that fasting states can change, for example, the lipids in the EVs. And so ideally, of course, all the healthy volunteers plasma was isolated from fasted individuals. The follow-up samples from patients were also fasted. But of course, when someone has a myocardial infarction and we take the acute phase, we can't look at fasting state. We have so essentially we couldn't really normalize the fasting states because these patients come in very acutely, and so that's not something we could really select for. But yeah, I think again, fasting is a great question. Yeah, especially looking at lipids. So thank you.
Okay, thank you. Thank you.
Thanks so much, Daan, for sharing this work, and to you and your team for publishing the first part of this. And we're looking forward to seeing what else is going to come out of this project. So this is really, really exciting. And Nazanin, thank you, and also Snev for helping to coordinate this session. I just want to say that if anybody has a paper that they would really like to see featured on here, we do have some slots in August especially, maybe one left in July. So please let me know. And otherwise, thanks to you all for joining today, and I look forward to seeing you on an upcoming EV Club. So thanks so much. Take care now.